I'm Dr Wihan Pheiffer from the PCDDP, or the Preclinical Drug Development Platform, which is a hosted entity at the Faculty of Health Sciences. We offer service research for companies and other academic institutions, both international and national, where we do preclinical testing on new medicines, vaccines, as well as plant medicines. Our research covers a wide range of topics, from investigating new plant medications to improve current drugs, as well as to investigate drug metabolism and drug interactions, all of these using animal models. My research specifically focuses on drug interactions and drug metabolisms. So, drug metabolism pharmacokinetics is when a drug is taken up into the body, how that is distributed, metabolized, and then excreted out of the body. And drug interactions are when any other compound interferes with this process. So the dose of the medication is either higher or lower than it should be. So the drug interaction research that we focus specifically on is herb drug interaction, and that comes from any medicinal plants that interfere then with this process. So an example would be if a person would take chronic medication and a dose is given at a certain level by your practitioner, and another alternative medication, which is plant medication, is taken, that might affect the dose of your chronic medication, which then either elevates the drug concentration, which can lead to toxic effects, or lower the blood concentration, that leads to no effect. So you're essentially taking a drug with no effects. Something that is not done in research that we are now focusing specifically on is inhalation of medicinal plants. So the smokes of certain plants is not studied on how this might affect specific drug metabolisms. So we have students who are now testing cigarette smoke and the effect thereof on chronic medications. So yes, smoke is a lifestyle choice, not necessarily for medicinal use, but how does that affect drugs that are taken by people every day? Does this lower the effect that you're getting? And we are using specialized models, animal models, to predict these changes. Now, these mouse models are changed in such a way that they express human genes. So the genes that we target are specific for those drugs that we're looking at. So if we are looking at either an anti-malarial drug or an anti-cancer drug or even an antiretroviral, the metabolism of that drug is then investigated using human responses in the mice. Potential studies that we're going to look at as well is if plants are smoked for depression, how will that interfere then with chronic medication? Because if it interferes with your antidepressants, then taking the chronic medication has no effect in the long run, which is overall bad for the patient. So the overall impact of the study that we're looking at is not just to predict clinical doses of the herb drug interaction and how that affects chronic medication. It also gives an indication of how overall health can be affected, because when a drug is administered at a certain dose and that interaction either increases the blood concentration, which results into toxic and adverse effects, it can also lead to the underdosing, and that underdosing then means that you're not getting the therapeutic effect that the drug is supposed to give.